afternoon, everybody. So I will be the first speaker at this session devoted to nanomedicines, the tiny tools to beat giant cancer challenges. And I'm a coordinator of the Maria Sklodowska-Curie-Rice project dedicated to integrative development of smart drug vector nanostructures for adaptive drug delivery into target cells. So first of all, I will show the slide about the global cancer burden. As you may see, so all the continents are affected. Mainly continent with the biggest amount of cancers reported is in Asia, followed by Europe and Americas. Well, our project involves research groups from 6 different countries. At the right hand, you see the European groups and our partners from the United States, Morocco, and Brazil. Namely, we have 7 academic groups and 1 big pharmaceutical company from Ukraine, which also participates the project. So the project is devoted to integrative development of those drug vector nanostructures. It means that we do in parallel at the same time synthesis and identification, isolation of pharmaceutically active compounds, either synthetic or natural. So we perform computer modeling of those compounds and their interactions with biomolecules. We perform biological testing, screening both of the compounds and the nanostructures. Then different approaches to the nanocarrier creation, synthesis of inorganic and inorganic nanomatrices, and stimuli responsive polymer nanocapsules. And all these those activities are realized through researcher mobility, either as exchanges with our international partners outside the European Union, as well as intra-European intersectoral exchanges. So the pharmaceutically active compounds, which we are dealing with, first go through the screening to see their cytotoxic activity and to evaluate their cytotoxicity both to tumor cells and to the normal cells. And in order to select the most promising anti-cancer active compounds to develop. There are several challenges in that kind of research, because we have to evaluate the efficacy of each particular molecule against its safety. Here I present some results from our project, how we are evaluating the compounds against different cancer cells. So several cell lines have more response to our treatment, to the active compounds we are using. However, there are several other lines which are not so sensitive. So here are the more promising compounds from our consortium, both synthetic and natural compounds isolated from Brazilian Amazonian plants. So those drug vector nanostructures, they are made in order to protect the active compound against the environment, both outside and inside the cell and the tissue, to decrease the degradation rate of those compounds due to moisture, light, oxygens, or enzymatic environment, to improve the workability of the active compound. So if it is liquid or oil, then it would be a solid form. Enhance bioavailability for purely soluble drugs, because we know that most of the anti-cancer drugs are hydrophobic. So they have quite pure solubility in water, and we have to enhance it. And chemical modification of those molecules often lead to enhancement of the solubility, but you have to pay with the decrease of the anti-cancer activity. So preparation of the drug vector nanostructure may provide a solution for that problem. So also the deviant development masks the taste and odor of the drug. And finally, we can provide the release of the active payload upon external stimuli. Here I would like to show a wide variety of existing nanoparticles and nanoconjugates from either inorganic and organic origin. Here in the left side of the slide, I will show our nanomatrices and nanoparticles prepared by the consortiums. It could be either of spherical shape or lamellar materials, or some polymer capsules. And those drug vector nanostructures are intended for targeted drug delivery. It means that we not only produce the particles of particular size, which could be delivered to the tumor site using the enhancement and permeability effect, because the vasculature in the tumor tissue is leaky. So these particles can just leave from the bloodstream to the tissue, and this is so-called passive targeting. As well as we are surface treating our particles in order to provide the molecules that could be recognized by receptors on the cell surface, and in such a way promote endocytosis of the particle inside the cell. So our objective is also adaptive delivery. If in the traditional approach we have small molecule drugs or encapsulated drugs, which may have some drawbacks like rapid clearance or premature degradation. So nanohybrid assemblies or drug vector nanostructures may provide a solution for that kind of problem. And especially we are interested in stimuli-triggered drug release by using several external stimulants like light, temperature, or change of the pH, acidity, or basicity in the tissue. So those are traditional systems controlled by internal stimuli, so by the properties of a particular material. And in that case, we are using the control by external stimuli. So we are trying to use the differences in pH and temperature within the tumor tissue and normal tissue. For example, the nanoparticles of thermal responsive amphiphilic polymers, which are developed by the consortium. Here you see the chemical composition of a polymer. It contains the hydrophilic and hydrophobic block. And this is the ratio between the two components. So the polymer particles are quite have quite narrow size distribution, but their solution in water has a property that upon a certain temperature, the polymer micelles starts to aggregate, and you can see this transformation upon slight heating. The solutions become turbid and the size of the particles increase. When you cool down the particles, so the solutions start to clarify, the micelles broke, and you have those small particles. Since the experiment was done in Italy, so the last sample remains turbid. If it would be in Finland, it would be clear also because of the lower room temperature here. So you may see the different particle sizes and diameters of the particles as a function of temperature. So when nanoparticles are close to the cell surface, they may enter the cell and be uptaken by endocytosis, and then secreted from the cell through the process which is called exocytosis. And all those processes are related to the cell secretory pathways. So this is the key component which we are addressing in our project. So different organelles which are make part of the cell secretory pathway. Upon entrance of a molecule or a particle inside the cell, then it goes to different organelles in order to produce proteins, polysaccharides, and their combinations. And they, in such a way, may influence the cell homeostasis. So in that slide, I would like to show you the role of secretory pathways in cancer treatment. This is quite new approach. So here are the differences between normal cells and tumor cells. Tumor cells have increased amount of endoplasmic reticulum, the elevated and folded protein response, endoplasmic reticulum Golgi trafficking, and strongly depend on different growth factors. So those parameters could be used to develop the drug vector nanostructures. As well, we are interested in immunomodulatory effects of our compounds and nanoparticles. So here in this slide, I show you the effects through the exposure of molecular patterns derived from different pathogens, apoptotic cells. And the short-term exposure leads to immunostimulation, and this is the case of acute inflammation. On the contrary, in tumor microenvironment, there is a persistent release of those exosomes, and as a result, the long-term exposure to those molecules lead to a chronic inflammation and to immunosuppression. So some examples of the action of drug vector nanostructures. So our project is dedicated not only to cancer, but also to neglected tropical diseases. And here is an example of how our system is compared to a molecular drug based on antimony, which is used against leishmaniasis all over the world. So efficacy of the nanoparticulated drug, you may see here in the right side. So the 100% parasite mortality could be achieved at 3 times lower doses of the active compound, which is highly toxic. The nanoparticles may also be efficient carriers for microRNA. And here is the example how the nanoparticles are prepared. Then they are characterized by size, by RNA release, and zeta potential. So depending on the ratio of the polyhydroxybutyrate and polyethylenimine, you may see here that zeta potential increases. That means that this nanoparticulated system became more stable in water solution. And finally, here you may see the influence of the forced expression of this micro RNA on prostate cancer cells proliferation and colony formation, which is also suppressed. So our experience with RISE project, we have both academic and industrial groups. So research meets the innovation in our project. So the fundamental science, which is the transformation of money into knowledge, and our industrial partners are participating in the innovation part, so in the transformation of the knowledge generated by the projects into money. The RISE project is a fantastic possibility to meet our and other colleagues with another background to make new contacts and friendships, to reinforce professional maturity of the participating fellows, to collaborate with eminent colleagues on different continents, and finally to cross-fertilize each other. Thank you very much for your attention.
So thank you so much, Andre. Apologies to everyone about my technology here. I think somebody turned on the washing machine and my computer crashed. But I'm back with the iPhone. Andre, that was a fabulous talk. Thank you for sharing your work on drug vector nanostructures, not only the knowledge that you're bringing to the field of nanotechnology, but your exploitative approaches for both cancer and also other diseases. And I liked how you mentioned in RISE about friendship, professional maturity, and collaboration. Maybe one quick scientific elaboration. With these drug vector nanostructures, which do you believe of your approaches is the most promising for cancer treatment. Over to you, Andre.
It strongly depends on the type of the tumor cell, as well as the material, because the goal is to develop the nanoparticle which could be more suitable for each particular cell. So I mean, probably for example, in organic nanoparticles, they are more active compounds as well. So this is the active nanomatrix, which contains both the drug and the carrier. Both functions may be performed. In the case of polymer material, we have a good vector for the drug and possibly more possibilities to tune the surface of the nanoparticle in order to be recognized by receptors on the tumor tissue.
Thank you so much for sharing your thoughts on that. So I see it's nanotechnology, especially the particles you work on, offer a lot of flexibility to tailor the approach to the type of cell and the type of need. It sounds really exciting field you're working in. I wonder now, maybe we can move on to the next speaker that I'll introduce, Miguel Castano. I'd like just to introduce him as a chemist, a physical biochemist. He did his PhD at the University of Lisbon, and he also is a mobile researcher. He did postdoctoral stays in Uppsala in Sweden and also in University of Hawaii. I'm glad we got him back to Europe from Hawaii. He's now the head of department in biochemistry, School of Medicine in Lisbon, University of Lisbon. And well, I didn't tell you, Andre is an active cook, a mushroom gatherer and history fanatic, and with his cat, whereas Miguel is into angling and sports fishing. But he won't be talking about that today. He'll be talking about his project Impact and how his project is developing approaches, also similar with a nanotechnology approach, but this time you're going to hear about peptides and how he uses them to deliver drugs to tumors that can reach the brain. So over to you, Miguel. I'm excited to hear about your work. Thank you.
Well, thank you, Amanda. Thank you for the nice introduction. After Andre, who told us about a more general approach to deliver drugs to different kinds of cancers, I will now address a specific kind of cancer, is breast cancer. It's a very aggressive cancer in some ways. And also it has frequent metastases in the brain and in the bones, and these are very dangerous metastases. So we have kind of a double challenge, which is the primary tumor at the breast and also the metastases. And we will focus also on metastases in the brain, because it's difficult to fight diseases in the brain because the brain is very protected. I must also tell that we have expertise on drugs that act on the membranes of cells, that act on the surface of cells. So these drugs, they destroy cells from the surface, okay? So they permeabilize cells, and they are actually good to fight cancer cells and bacteria. This may seem awkward in the beginning, as bacteria and cancer cells cause very different diseases. But from our point of view as biochemists, I mean, from the molecular and cellular point of view, they act in a very similar way. To address such an interdisciplinary approach, okay, we decided to apply for RISE, because RISE is actually very adequate for interdisciplinary teams, interdisciplinary approaches, so quite adequate for innovation. I hope my presentation is going on. Okay, so I will just tell you briefly, I will just show you briefly a very short video that tells about our project, how it is the project in general, and then I will tell you about the results we got on breast cancer. Okay, but now what is our project, how it started, how was it built, what was the opportunities that it created? It will show in this video, hopefully it will go well.
IMPACT aims to develop a platform technology to produce innovative peptide-based drugs that will be delivered to pharma industry for development and future clinical applications. The unique character of this platform results from the involvement of an international team, including 5 academic partners and 3 biotech enterprises committed to research and development. Together, this team combines specific technologies and expertise, and will work at the interface of basic and preclinical research to translate the outcomes into technologies of industrial value. This platform technology will be first used to develop drug leads for brain tumours and infectious diseases caused by a pathogenic bacterium, Staphylococcus aureus, for which current therapies have limited effectiveness and/or are susceptible to the development of resistance. Indeed, on the one hand, brain tumours are not accessible to chemical drugs, with only 2% of the known drugs able to traverse the blood-brain barrier. On the other hand, Staphylococcus aureus has multi-resistant strains and can grow in the form of biofilms. Bacterial biofilms are hard to eliminate, as most antibiotics are unable to diffuse across and/or destroy the extracellular matrix, being therefore prevented from reaching bacteria. In order to tackle these two challenging unmet medical needs, IMPACT aims to develop new generation drugs by producing chimeras that combine anti-tumoral or antimicrobial molecules with small domain antibodies and/or barrier translocating chemical entities. In addition to the research and development partners, IMPACT also involves a world leader European business school, an established media partner specialised in science communication and experts in rising research funds from the Stanford University. This consortium has the perfect environment and facilities to develop and train early career researchers. Students will therefore have the opportunity to acquire knowledge and skills in science, technology, entrepreneurship, business, and communication so they can proactively tailor their career path and a lifelong learning perspective.
So this was the context that we have set, a very interdisciplinary and intersectoral team. We also wanted to address communication and actual translation to the business world, and Bryce gave us this opportunity. So, but in this presentation, I will tell you about the results, not all the results of the project, but the results concerning cancer and metastatic breast cancer. So our aim was to fight breast cancer. More than that, fight metastasis in the brain. So what was new that we wanted to do? Well, we wanted to explore a new biological drug. This means a small protein that actually exists in nature, specifically in common bin. Okay, so this new protein, we knew it had properties that could be used as a anti-cancer drug. And the strategy is to kill cell membranes and at the same time prevent metastatic cancers to install in the brain. Okay, so why membranes? Why do we want to use a drug that actually kills the cells from the surface? Okay, by destructing the membrane that actually isolates the cell. Well, it's because there are lots of different aspects concerning cancer. There are lots of different drugs, okay, in these boxes around here that address these targets, that address these characteristics of cancer cells. So there is a wide variety of different drugs to actually fight cancer. And of course, if you have drugs that address different targets, they are potentially additive on their action. So you can add the effect of different drugs if they address different targets. And you see that among the available drugs, none is actually targeting the membrane. So we could bring into this play, okay, a new player, a new drug that actually addresses a new target. And so you are actually adding value to the context that you have now in anti-cancer drugs. So that was our motivation. As I told you, one big problem about breast cancer is that it develops metastases very easily in the liver, in the lungs, in the bones, and in the brain. Each of these metastases is quite dangerous and quite difficult to address. But probably the most difficult one is the brain. But we also worked in other lines trying to find peptides that cross from the blood to the brain. So they are transporters of drugs from blood to brain. And so we thought we could combine actually both properties in a single drug. But the project evolved. I will not go very much into details. But of course, if you want to kill the cancer cells and prevent metastasis, you have to think about the biology of cancer. So you have the primary tumor. Some cells release from the primary tumor, they enter circulation, somehow they adhere to the surface of arteria or veins somewhere. They install there and they start their own community. So they start a new cancer there. So if you think about breast cancer and brain metastasis, what happens is that you have the primary tumor in the breast. Some cells are released, some cells enter in circulation, and then in the brain, in the arteries of the brain, they actually glue or they can transverse the arteries in the brain and actually install in the brain and start a metastatic cancer in the brain. So if you want to address all this problem, what is it that you have to consider? Well, you have to kill the primary tumor, that's for sure. That's where the problem is primarily. But also you need to prevent metastatic cells from actually finding a place to create a community or actually kill the metastatic cancer. That's what we try to do. We try to address this problem this way, taking into account the biology of the cancer. So this small protein we extracted actually from the common bin. It's a defensin. It works as an antimicrobial in the common bins. Okay, it can bind to the surface of cancer cells in the breast and actually kill cancer cells. But we also measured how this molecule could interfere with adhesion between cells, the adhesion between cancer cells and artery cells. Because to create a metastatic cancer, of course, cancer cells have to actually contact and adhere, kind of glue to the artery cells to at some spot create a new cancer, a new community. And it's possible to measure the adhesion forces, the interaction forces between cells in the absence and in the presence of this new drug we are studying. So we studied the capacity of this new molecule to actually reside at the surface of the artery walls in the brain and prevent metastatic cancer cells from adhering to this surface. So from gluing to this surface, therefore preventing the installation of metastatic cancer there. So that's what we did in one of the phases of our study. But of course, as you understand, you don't need to be an expert in this. I mean, using kilos and kilos of bins to extract a few milligrams, it's not very cost effective, and it's very laborious, also very tedious. So if we want to make it a real therapeutic alternative, we have to synthesize the molecule and not rely on extracting the molecule from the bins. Okay, we have to devise a way to produce large quantities in a cost-effective way and quite automatic way. So our partners that are expert on synthesis, and they were part of this project impact in this RISE project. Okay, they have devised a way to chemically synthesize the molecule, although it's a relatively complex molecule. It's a small protein with disulfide bridges in between. We have tested the molecule for its natural function in a live model, in a worm. Okay, but we have managed to produce the protein in large quantities, and we were quite happy and quite satisfied with that. Okay, so this is a comparison of the structure of the molecule synthesized and then isolated. These graphs here, they tell you that as you can extract the molecule from the natural source, the bin, can isolate the molecule, but you always have some unwanted molecules coming along, of course, because you are isolating these molecules from a plethora, from an enormous quantity of other molecules. While if you do it synthetic from chemical synthesis, of course, you can isolate the only molecule you want. You have it highly purified, which is from the pharmacological point of view, much better than using a mixture, of course. So just this was our work in this field in a nutshell. Of course, I have another video because we use videos to kind of outreach and promote our results, because we want to go far behind the walls of academia and try to reach the common citizen, and trying to explain what we do with taxpayers' money, actually. So we are always concerned in kind of presenting our results in a way that everybody can understand. And out of curiosity, I will show you a video too that explains the outcome of this project in this field.
Breast cancer is a malignancy most commonly diagnosed in women, and the second leading cause of cancer-related death in women worldwide. As the therapeutics targeted to localized masses in breast improve, metastatic lesions in the brain slowly increase in their incidence, compromising successful treatment outcomes overall. The treatment of brain metastasis by chemotherapy is, however, further compromised due to the inability of therapeutics to penetrate brain arteries, known as the blood-brain barrier, and act on neural tissues. Indeed, in addition to the low passive paracellular permeability, the blood-brain barrier cells express high levels of drug transporters that mediate drug efflux, and consequently the brain is a protected location for malignant cells to settle and proliferate. That is why developing methods that can result in improved blood-brain barrier penetration of drugs and their availability in brain sites is of utmost importance and an urgent necessity. Peptide-based therapies have emerged as the pool of new strategies capable of overcoming unspecific targeting and resistance issues. In this work, we reveal the anti-tumor activity of PVD1 peptide, a natural antimicrobial plant defensin from Bacillus vulgaris, the common bean, on human breast cancer cells, and its ability in aiding the prevention of brain metastasis formation by affecting cellular adhesion to brain arteries. We have used a combination of techniques that probe PVD1's effects at the single cell level and reveal that this peptide can effectively damage breast tumor cells. Our results suggest that PVD1 quickly internalizes in cancer cells, but remains located in the membrane of normal cells, with no significant damage to its structure and biomechanical properties. These interactions in turn modulate cell adhesiveness between tumor and blood-brain barrier cells. Taken together, our results show that PVD1 is a potential template for the design of innovative pharmacological approaches for metastatic breast cancer treatment. The manipulation of the biomechanical properties of tumor cells that ultimately kill the tumor and prevent malignant cells to attach to the blood-brain barrier.
So if you're keen on more scientific details, you can actually consult the 3 papers that have been published about this. I'm glad to also interact with anyone that wants more information on or more details. I guess the general picture of the project was this one I gave, but of course you are welcome, more than welcome, to consult the papers that we have published. Thank you very much for your attention. I'm willing to take questions in the end or now, whatever. Thank you.
So I guess it's so over to me. Thank you so much, Miguel. I'm really just absorbing what you've told me. I'm quite fascinated. It's kind of killing 2 birds with 1 stone. You found a peptide naturally occurring in beans. So this is inspiring me for lunch, that's for sure. And these peptides can kill tumor cells and prevent attachment. I think you've shown us that you've not only come up with a nanotechnology approach to target cancer cells, but you showed us how important it is in the field to work in teams, to understand the biology of cancer so that chemists can dream up these wonderful materials and exploit the properties of materials. So, and thank you for showing the videos and how important scientific communication is to help us appreciate your work. So just a very modest question before we move on to Yelena is these peptides, the so-called PVD1, how does this compare to currently available therapies? What's your opinion on that?
Okay, we try to combine the best of 2 worlds, because nowadays you have what we call small molecules. They are artificial molecules, easy to synthesize, but they are quite unspecific, and they are quite toxic, and they target all sorts of cells that divide rapidly. That's why so many anti-cancer drugs cause air loss, okay? Because air grows permanently means that the cells of air, they divide permanently. And so when you try to fight cancer with very unspecific drugs that target cells that multiply a lot, you have air loss, and that is a problem. So on one hand, you have small molecules, easy to synthesize, but very unspecific and having lots of toxic effects. On the other hand, you have antibodies, which are very specific, but they are big molecules, they are big proteins. Sometimes they are difficult to deliver. You can use the methodologies that Andrew uses to deliver drugs everywhere. So that's one way to address the problem. The other way is to use proteins that are much smaller than antibodies, but somehow are specific, and being biologic do not present the same kind of toxic problems as small drugs. So we try to combine what is best in antibodies that are now being used and small molecules that are also being used, and try to gather the best, the qualities of both types of drugs, and actually reduce the side effects. So we are trying to find a third alternative to address cancer with biological drugs other than antibodies.
No, thank you for explaining that. Sometimes when you're not an expert deep in the field, it's hard to know what's one approach better to another and where should funders invest their money. But I think what you're showing is how scientists need to really understand the field from all perspectives.
Yeah.
And as you said, take the best of each approach and combine it into what you've shown us is a very novel approach. And I'm really thankful to hear that that's underway, and I will definitely be following further your project and encourage other people to do the same. So thank you very much, obrigada. We're moving over to Yelena, and I've just noticed all my papers are all over the place. So how do I introduce you, Yelena? I might have to rely on your own introduction. We did, so Yelena is working, as she said, in Stuttgart now. She could tell you a little bit more about her impressive background. She's starting out as a physicist, and she's worked her way through into the medical field, and impressively now in Max Planck in Stuttgart, in a whole new world. But for me, she's a role model for women in science, and also for anyone in science, crossing borders and working in these teams to bring her knowledge to this field. And today I've asked her to talk about the project she's involved with called MAGNAMED. It's also a RISE project. That's the theme today and connection of the projects. And this project explores the design of novel magnetic nanostructures and their unique properties. Again, we're going to look a little bit more at diagnostics and also improvement of therapy. Yelena, I'll pass it over to you. I just want everyone to be not only impressed by your science, but also I hear you're a sailor and a person who flies. So I think someone with this profile really is going to lead the way in new discoveries that will help us fight cancer. So over to you. Excited to hear about your work.
Thank you so much, Amanda, for a very kind introduction. So as you said, I'll be talking about the RISE project with the acronym MAGNAMED and introduce magnetic nanostructures for medical applications. And 1 second, I need to figure out how to move on. Okay, so my previous position, we were partner of MAGNAMED, and I've worked as a head of preclinical imaging facility in Vienna that was located in Vienna Biocenter in Austria. And here are the two ladies that worked with me together on this project as well. So we were very lucky to be one of the only 4 places in the world to have 15.2 Tesla state-of-the-art magnetic resonance imaging scanner. And as I said, we are only the partner. The principal investigator on this RISE project was Rafael Morales from, he is a professor at Universidad del País Vasco in Spain. And he actually, I would say, he really inspired me to move on with this project. And I have to thank him. All the contributions should actually go to him. So basically, the MAGNAMED project, in a nutshell, is the center of the project is the nanofabrication of a permalloy disc, nanodisc, that can be in the vortex magnetic state or antiferromagnetic state, depending on the application. So once we produce this nanodisc or nanostructures, we can functionalize them, and they can serve as biosensors to detect very small amount of proteins in the blood, basically a tumor markers, so to lead to very early detection of the tumors. Or we can functionalize them, and they can be used to be actually with the functional proteins, they can be targeted to the tumor tissues, and there we can use them to induce tumor hypertermia. Now I have to emphasize here that the tumor hyperthermia, the mechanism of magnetic nanostructure is very different from what I'm going to talk today. And basically with magnetic nanostructure, you're using the oscillating magnetic field. And talking to Rafael, we actually came up with the idea to, instead of magnetic nanostructures, to produce gold nanodiscs. And it will be clear why I wanted the gold nanodisc instead of the magnetic nanostructure in the next few slides. So basically, as I said, we are 15 partners with very different expertise from physics, chemistry, biology, and medicine. So basically, I think it has their own contribution to the success of this project. So just a brief introduction to the MRI. The MRI has many parts, some of which are hidden, but basically the main part is this big magnet. This is a static magnet, so the magnetic field cannot be changed. It's always on. It has inside the gradient coils, which are basically magnetic field gradients, and they serve for the formation of 2D and 3D images. And they're also the RF coils. So basically the RF coils are the center of what we are going to use for the hypertermia against the cancer. And on this image, you can see the coil, it's here. The mouse will typically go inside the coil, and this coil can generate radio frequency field, which can be very powerful. And we are going to use this radio frequency field to induce the mild hyperthermia. Since the magnetic field strength is 15.2 Tesla, the Larmor frequency is 650 megahertz, which is actually quite high. Now, the motivation, why would we switch from magnetic particles and actually start using the conductive particles and actually use the MRI for the hypothermia instead of just the oscillating magnetic field? First, MRI is non-invasive imaging modality that is already commonly used in clinics for diagnostic purposes. So this is the MRI image of a mouse abdomen, and here we can already see the tumor. And now, wouldn't it be nice if we can add some minimally invasive treatment as well? And the advantage is obvious. So the advantage would be to image and treat in the same basically imaging session. And we also decided to focus on the breast cancer. As Miguel said, it's the most common cancer in women. And also for this proof of principle was perfect because it was actually outside the body. I think it would be much harder to treat something that's inside the body, but this is not the topic for today. And yes, so the question is, can we now accomplish hypothermia by basically injecting the mouse with the small gold nanodiscs? So the principle, why would this be possible, is that electromagnetic field or the absorption of electromagnetic field can lead to heating, and this is not new. As a matter of fact, during each clinical scanning, something that's called specific absorption rate is strictly monitored, and the FAA has the strict rules for the SAR rate during the MRI scanning. So this is the power basically deposited into the tissue that can lead to heating. And this heating is unwanted heating because you don't want to heat the patient and have like the skin burns while you're performing the MRI. This specific absorption rate actually depends on the tissue conductivity, depends on the electrical field in the tissue that's been coming from actually the RF coils, and it's inversely proportional to the tissue density. And basically during the MRI, body is exposed to both electric and magnetic fields, but for the hypothermia, we actually only care about the electric fields. So our initial starting point was numerical stimulation just to see whether the idea is worth trying. So what we simulated here is the RF coil that you see the outside, and basically a cylinder that is filled with saline agarose. So basically we model the conductivity of saline. And on the tip, we are putting the increased conductivity, which will come presumably from the gold nanoparticles. And as you can see from numerical simulation, it seems like that increase the specific absorption rate is where you have the gold nanoparticles, nanodiscs. So before we move to the real treatment, we spend, I think, quite a few years studying the tumor vasculature. And I would say that really knowing the tumor vasculature is the key for the cancer treatment. And you will see in a minute why. So in order to treat the cancer cells with any kind of drug, you need a way to deliver the drug to the tumor cells, and vasculature actually represent the natural conduits to the tumor cells. However, the tumor vasculature is very, very different than the normal vasculature, so we need to take that to work in our advantage. So here you can see in this animated movie, you can see pseudocolored in green tumor and one small vascular tree, and the rest of the tumor seems to be devoid of the vasculature. However, if we perform MRI scan where we can quantify the tumor blood volume, we can see here that the tumor blood volume is quite increased compared to the rest of the tissue. So if we segment this tumor, we see different small little vessels. But if we now change the modality and use optical coherence tomography, we can see that there are very, very tiny small vessels all over the tumor. So in conclusion, the tumor vasculature comprises of very small blood vessels that are less than 15 microns, so they are pretty much invisible for the MRI. And now I'll have to switch to the next slide where you will learn about additional properties of these tumors, why they are very good candidates for the hyperthermia. So there are two main things. One is that tumors are low perfused compared to the normal tissue, which contributes to much slower cooling. Here is the brain of the mouse. And again, the breast cancer, this time located near the neck, but I'm just using this to show you that compared to the brain where perfusion is quite high, these tumors are poorly perfused. So if we are to inject mouse with the nanodiscs and try to heat this, just the regular blood delivery to the brain will naturally cool the brain tissue, while this tissue will not be cooled. So there is a higher chance to heat this tissue. And the second is what Andrea already mentioned, it's enhanced permeability and retention effect in tumors. This means if you're looking at the K2, which is connected to the permeability, these tumors have increased permeability compared to different tissue, which means drugs can easily penetrate inside the tumor. And as I mentioned already, they have increased tumor blood volume, which means that, there will be a lot of drugs accumulated in the tumor. But now let's look at the really enhanced permeability and retention effect, at what that means if we are injecting the mouse with the drug. So for these purposes, and actually this was to determine the time when is the best to perform hypothermia treatment after we inject mouse with the gold nanodiscs as well. So gold nanodiscs are not good contrast agent for the MRI. So for these purposes, we were actually using 17 nanometer iron oxide multi-iron. So this is a very good contrast agent that's contributing to the reduction of transverse relaxation, or the T2. So what you're looking here are the quantification, the measurement of the T2 relaxation time at the beginning before any injection, and then at 15 minutes, half an hour, 1 hour, and so on. And when this tumor starts to become blue, that means that there is a lot of iron particles accumulated inside the tumor. And we are concluding that based on the reduction in the transverse relaxation time. So if you're looking at these graphs here, you can see for the normal tissue, the red line, there is actually pretty much no accumulation in the tumor. The magnetic particles come in and they're being cleared pretty rapidly, while there is actually accumulation with certain delay, which we determined to be around 6 hours. And this is the time when we are going to treat the mouse with hyperthermia when the hyperthermia treatment will be performed. And then slowly these particles are being cleared from the tumor. And yeah. So in the next slide, just to briefly describe the nanofabrication process, which was done actually with the help of our partners. So we were basically producing the gold nanodiscs, which are 4 micron in diameter and 100 nanometer thick, using the lithography. And here is the image from the Shore lab in University of San Diego, actually the actual instrument that we use for the nanodisc fabrication. And on the right side is the electron scanning microscopy image of the gold nanodiscs, also done at the University of San Diego. So the MRI has additional advantage, and that is that we can actually measure the temperature using MRI. And the physics is not so complicated. Basically, the resonant frequency depends on the temperature, and there are 2 terms. One is the magnetic susceptibility, and the second is this electron shielding of the nuclei, which is really dependent on the temperature, and it's contributing to the shift in the frequency. So basically, we can look as change in the temperature by measuring change in the phase. And we can do 1 phase image before increasing the temperature and 1 phase image after, and we can actually get relative temperature increase. So for the ex vivo studies and the proof of principle, we had Eppendorf tube filled with the gelatin, so the agarose gel dissolved in the saline, the physiological saline, and here are nanodiscs, so the gold nanodiscs. And once we expose this to the 2 minutes of the RF pulses, and I should say here not continuous, but every pulse was about 5 microseconds, and repeated every second. We are doing this to avoid any damage to the MRI equipment. We can see that there is increase in temperature in about between 5 and 8 degrees where these nanodiscs are located. And just to describe our breast cancer model, so we are using syngeneic models, so we are injecting the mouse breast cancer cells. This is how it looks in advanced state, I would say, after 3 weeks following injection. So we are letting tumors grow about 5 millimeters in size. And then we start doing MRI daily to monitor the tumor size. And we're trying to treat before there is the necrosis inside the tumor. So here is just a regular MRI image. The anatomical location of the tumor is here. And when they're about this size, we are starting the RF hyperthermia. And we can do RF hyperthermia, it takes only 2 minutes to do it. So for that, we use the rare imaging sequence, but we are increasing the power of the pulses. So it's 60 watts for 90-degree pulse and 80 watts for 180-degree pulse. And as I said, these pulses are about 5 microseconds in duration. And we also have the optical thermometry, the temperature probe based on gallium arsenide crystals, which is located underneath the mouse tumor so that we can measure the temperature during the whole treatment, not to exceed, I would say, ever 50 degrees. So with our MRI thermometry, we can look at the phase maps before and after, and we can measure the temperature inside the tumor and conclude that there is increase in temperature so between 2 and 5 degrees following the hypothermia treatment. And now, how does this work? Here is the picture of a mouse that was imaged at 0 hours, and then after 48 and 72 hours. And as Miguel mentioned, the breast cancer tumors, especially these, are very aggressive, and you can see that there is almost doubling within 72 hours, which means that there is an exponential rate of growing of these tumors is reached. However, if you look at the tumors that was treated with nanodisc and hyperthermia, the RF hyperthermia, you can see that tumor is shrinking in size. So after 72 hours is even smaller compared to 48 hours, which means that the cells that are dying from the RF hyperthermia, it's actually a slow process. So they are not being killed instantaneously. And if you're looking at the basic H&E histology, we can determine that there is a lot of necrosis, especially here, which appears with the scarring tissue, and there is a lot of hemorrhage. And I would say that the mechanism of treatment is both destruction of blood vessels as well as destruction of tumor cells, which is kind of novel because you're working now on both ways. Usually tumors either destruct blood vessels with anti-VEGF therapies, or they only destruct the tumor cells. And this is just an illustration how it looks on a mouse. So this was before the treatment, and this is after the hyperthermia treatment. You actually cannot even see the tumor. Tumor is inside the skin where it used to be. Now, in conclusion, I think we demonstrated a proof of principle that hyperthermia is effective against breast cancer. And it can be achieved using MRI. So these are very, very preliminary results in the presence of gold nanostructures. But I think the next step will be to actually go back to the MAGNAMED and use this in the presence of magnetic nanostructures. I think basically based on our numerical simulation, it might be even better. And the possible future work will be if we can explore MRI gradients, we can actually move magnetic particles to targeted location. And we are already doing this at Max Planck Institute. We can also basically do some improvements in the RF antenna design for more local and focal, stronger RF field delivery. And we can also exploit robotic delivery of nanostructures as our nano and micro robots and also biohybrid robots. I would have to thank everybody, all the partners from the RISE project. They have been wonderful. I think this was like one of the most fascinating research that I've done in my career. Yeah, I would say big, big thanks to both Ivan and Rafael for including me in this project. And of course, big thanks for European Union for funding the project. And thank you all for your attention.
Hi there, it's me back again. Wow, Yelena, thank you so much for making this understandable, your work. It's actually, I've never heard of the idea of using hyperthermia for treating cancer. I know about hypothermia because living in the Nordic countries, it's quite cold. But seriously, could you elaborate on how tumor hypothermia treatment compares to other treatments? Miguel had explained his approach is less toxic, and I picked up yours is less invasive. It combines this monitoring function with the treatment. But could you help me understand more how this treatment compares to other cancer treatments?
First, I would say what Miguel is doing is really fascinating because he's even trying to prevent metastasis, and brain metastases are a huge problem. And regarding my hypothermia treatment, I think the trick is to destroy both blood vessels so the tumors do not have any blood supply, and also to destroy tumors as well. And I would say that since the tumors are highly acidic, that probably there is slightly better RF power absorption in the acidic environment. So we are kind of doing both destruction of blood vessels and destruction of tumors at the same time. But since this is very early stage, I think lots and lots of further research will have to be done that this is really localized and that you're not affecting other tissue as well. So.
No, thank you for explaining that. So it seems maybe we can bring everyone back together because it seems you have a lot to talk about with each other's approaches. It seems you could make a new RISE project together, combining all approaches. But if the experts at ESOF could help us all talk at the same time, I could moderate question and answer session. We can take some scientific questions or more policy oriented questions. I have a few prepared. And also I welcome the audience to put some questions in the chat box, and I will try to address them with the colleagues. I also have received a few questions by email by chance. So I have lots of questions. We have just about 20 minutes to discuss and come up with what we've learned today. So is everybody in the panel discussion now, or can we all see each other? Let's have a look. I've got 5 computers up. I see me and Yelena. Okay. So let me start. So I guess scientifically, well, what I've learned today is that it's not enough just to be creative in the laboratory of making using nanotechnology. You really need to understand the biology of cancer, of different cancers. Yelena was with the vasculature, with Miguel, the mechanics of metastasis, and Andre also showed what he needs to know to tailor his particles. So maybe a question to all of you to discuss, what are then the main challenges in the field? How will you move your novel discoveries further to be applied in actual clinical trials and eventually treatment? So maybe you could discuss together the challenges, how you hope to overcome them, any policy areas that could help you overcome them, or scientific. So why don't you chat together, and then I'll bring in some questions about the type of scientists you need. So first, about the challenges. Who would like to take the floor first? So just unmute and jump in. How about Miguel? Would you like to comment?
Yeah, I can comment. I guess the classical problem, it's not new, and it persists over the year. It's I mean, for people that research on the basics, on the fundamentals of the processes, the problem it's always to jump from in vitro or in vivo and model and tests in animal models into humans. I mean, this gap, it's identified as a giant gap, as a very difficult to actually overcome. But somehow we persist in discussing that we need alternatives to bring the so-called basic science. I don't like calling it basic science, but anyway, bring basic science closer to the first in human tests. And somehow over the years we discuss, but nothing happens. And this is where natural science meets industrial application and meets policy, actually, because there's a lot of policy in actually being able to test in humans. And of course we have to do it the safe way, but there has to be an easier way actually to transpose this valley between in vitro and animal testing and first in human tests. I guess for most of us, I don't know, Elena will say and Andrew will say, for most of us, the main difficulty is actually not taking the projects as they are. It's actually going behind the project and reach out for a larger scale. I mean, we can contact with biotechnology people that are doing the preclinics, but even for biotech companies, it's difficult to then reach out for human tests. because of regulation, because of investment. I mean, it takes a lot of money to being able to meet all the requirements for tests in humans. We should all be looking for, of course, safe, but somehow easier ways to reach for human tests, because it's also known that animal tests don't tell you everything, are not all that reliable, as most reliable as you can get, but they are not totally reliable. So you may lose a lot of molecules and processes because they have failed in animals, and who knows, they probably could work in humans, and the reverse. I mean, it's also because there are lots of molecules and processes that work in animals, and then they are a total failure in humans. So this, I would say, is our most biggest concern.
No, thank you for mentioning that. Indeed, I think 2 points I heard is moving from animal models to human models and trials. But if I heard correctly, the regulation, the safety issues, the ethics, it seems there's a lot of barriers then. And maybe being involved with RISE projects helps train scientists to know what they have to do to use their, we call it explorative research, to move it into the exploitation stage. But maybe can I ask to Andre and then to Yelena, how do you think of what Miguel explained as the challenges of this animal to human and the other issues? How have you experienced these challenges and how do you overcome them? Andre?
From my point of view, we have a lack of an adequate regulation for nano-formulated drugs. So the current regulations, they are specific for molecules, either small molecules or big molecules, but not the nanoparticulated systems. Since we are in contact, we have in consortium a pharma company, a big pharma company, so they are quite concerned about the nanostructured drugs, which are don't well regulated. And another question is that for us, as for chemists, sometimes there is a challenge to go through all the preclinical steps from in vitro to in vivo, because many in vitro tests include particular cell culture media, etc., developed in such a way that nanoparticles became not stable. So we have, as I told, a half, let's say, of our project is also devoted to neglected tropical diseases. So we saw that sometimes our drug vector nanostructures simply fail in vitro tests, but they are doing pretty well in animal tests because of this incompatibility with cell culture media. So it means for us that in order to show good in vitro results, we practically have to develop another type, a special kind of a particle, especially for that purpose, that will not go otherwise. And will not go further because it is only for in vitro. This is the biggest challenge maybe for our project, because we are just at the beginning, at the preclinical step. But it is very important that our consortium includes chemists, biologists, immunologists, and physicians. So it is a very good exchange of ideas, and it's a good place to acquire new knowledge and to be acquainted with the specific problems of the other field.
No, thank you for that answer. So you've added indeed nano safety, if we could call it that.
Yeah.
That we're lacking perhaps regulations for nano drugs. It's okay maybe with molecules, and then also animal regulations that clearly the work you have to do has certain steps till you even go to an animal model, let alone a human model. And then this can also slow down research and create barriers. So I think it's great how you approach that with the industry input. I'm just looking at my clock. I see we have 5 minutes more, but Yelena, do you have a few words to add about these scientific challenges and how policy could overcome these, or what you think about it briefly?
I would just comment on what Miguel said, the difference between animal and human research. It can be especially challenging when you're modeling drugs because it is known that animals have different drug kinetics and also different drug metabolism. So what might work, basically what Miguel said, what might work in animals does not necessarily work in humans and other ways around. And what I would like to suggest for, since I am getting more deeper into the nanofabrication, nanostructure, robotics, is that we really should be looking at something that's biocompatible and also biodegradable. So nobody wants these magnetic structures deposited in their body. And I think FAA, even like probably 10 years ago, banned the use of contrast agent, these paramagnetic particles, because they could not adequately clear from the body. So they were being deposited in the liver and were staying in the liver for quite some time. So those are the things that we should really be looking into. And yeah, the policy are totally different set of challenges on their own. I think they're there mostly to protect us.
Yes, no, thank you again, all of you elaborating on these clear challenges that policy needs to hear from you, what the challenges are and how to overcome them. So it's very helpful what you've mentioned. Thank you for that. So we have 3 minutes left. While I have Yelena there, just a quick question that came in the chat was about the temperature that people could stand with the hyperthermia. How can you give us a feeling about that? And then I'll close the session. So tell us.
Sure. So as I said, the tissue itself has the blood vessels and it's a very efficient way of cooling the tissue. So even if you're to heat tissue briefly to 40 degrees, or let's say 45 degrees, the blood will come immediately and it will immediately cool the tissue down. So it's about really being exposed to prolonged hyperthermia. I would say more than 5 minutes. And here we are using the advantage in the cancer tissue that the perfusion is really slow. So the tissue is not being effectively heated. And what I was doing at the beginning is I would keep the mouse under the cold blanket just to prevent heating, which later on turns it wasn't necessary because I wasn't really heating the tissue. So that's it.
No, thanks for that trivia. I see we're running out of time. An excellent question came in about how not only to have engineers and physicists and molecular biologists involved, but how to get medics involved. Well, I guess that helps me in wrapping up the session, because I think what I'll close is to tell you, I hope everyone listening has been inspired today by what we've heard to apply for RISE, because you can work with all the different disciplines and all across the world. In the last seconds, I thank everyone who was involved today to prepare, and thank you speakers. Everyone, contact them, read their papers, and have a look on Twitter to see the outcome of our last call. We had 74 projects, 823 organizations, and over 100 SMEs involved, and almost 150 countries. So thank you, everyone. I think we've run out of time. And I wish you a wonderful rest of the day and good success to all the scientists today to further your career and your scientific work. Thank you so much for your time and your collaborations.
Thank you, Amanda.
Thank you.
Thank you, Amanda, and thank you, the organizers, for the wonderful meeting.
Yes, thank you, Andre. Now back to our hobbies. Yeah, so have a lovely afternoon, and I will share the other questions that came in to you, so in case you'd like to answer them in your future communications.